In 1665, the residents of a small English village made a decision that most people would not make even with a gun to their head. They chose to stay inside a plague outbreak and let it kill them, rather than run and risk spreading the disease to everyone around them. About 260 people died. The village was called Iam in Derbyshire, and it was never more than a few hundred people at its largest. But 350 years after those deaths, scientists studying HIV, the virus responsible for the AIDS epidemic, found something in the blood of IM survivors' descendants that stopped them cold. A genetic mutation, one that blocks HIV from entering human cells almost completely. And when they traced where that mutation came from and why it existed at the frequencies it did in European populations, the trail led directly back to the plague, to the people who died in it, and to the people who somehow survived. This is the story of IM, of the Black Death, and of the single gene that connects a 17th-century village quarantined to a possible cure for HIV. IM in 1665 was a quiet lead mining village in the Peak District. Estimates of its population before the plague range from 350 to around 700, with most historians settling on approximately 700 based on parish records and tax rolls. It was connected to the outside world mainly through traveling merchants and traders, which is exactly how the plague arrived. In August 1665, a bale of cloth came to Iam from London, a city then in the grip of the Great Plague, which would ultimately kill approximately 100,000 Londoners, roughly one-quarter of the city's population, over 1665 and 1666. The cloth was addressed to the village tailor, Alexander Hadfield, and was handled by his assistant, George Vicars. The cloth was damp from its journey. Vicars laid it near a fire to dry it out. Within a week, George Vicars was dead. He died on September 7, 1665, the first plague victim in Iam. The mechanism was simple and brutal. Bubonic plague is caused by the bacterium Yersinia pestis, transmitted primarily through the bites of fleas that have fed on infected rodents. Fleas and their eggs can survive for weeks in cloth and organic material. The damp London cloth, warmed near the fire, released infected fleas into the Hadfield household, and the outbreak began. Through the winter of 1665 and into the spring of 1666, plague deaths in Iam accelerated steadily. Families were dying, neighbors were dying, and many villagers had begun to do the obvious thing: pack up and run. At this point, two men changed what happened next. The village's rector was Reverend William Mompesson, just 28 years old and newly arrived in the parish. His predecessor, a nonconformist minister named Reverend Thomas Stanley, who had been removed from his official position by the Act of Uniformity, but still held enormous moral authority among the villagers, worked alongside him. Together, these two men persuaded the community to do something that has almost no parallel in the history of epidemic disease. They voted voluntarily to stay. The decision was not forced on them. There were no soldiers at the border. No one was physically prevented from leaving. The villagers of Iam looked at what was happening to their neighbors, understood that fleeing would spread the plague to Sheffield, to Bakewell, to every market town within walking distance, and chose collectively to remain inside the infected zone and accept whatever came. The boundary of the quarantine was marked by stones at the edges of the village. Villagers agreed on their honor not to cross them. The outside world agreed to deliver food and supplies, organized by the Earl of Devonshire, who supported the quarantine and arranged regular provisions, left at pre-designated spots at the village perimeter. Payment was made in hollowed-out stones filled with vinegar or running water, both believed to neutralize contagion. Vinegar was a standard early modern disinfectant. Reverend Mompesson moved all church services outdoors, preaching in the open air at a natural rock formation now called Cucklet Delph, to reduce the risk of transmission in enclosed spaces. He organized the burial of the dead, often requiring families to bury their own members without communal assistance to prevent further spread. His wife, Catherine Mompesson, died of plague on August 25, 1666. Mompesson himself survived. The toll was devastating. The most commonly cited death count is approximately 260 people, out of a population of around 350, which would be a mortality rate of 74%. making IM one of the most catastrophically affected communities in English plague history. A more recent epidemiological analysis published in 2016 in the Proceedings of the Royal Society B suggests the pre-plague population was closer to 700, which would place total deaths at around 257, still approximately 37% of the village, an extraordinary loss by any measure. The Hancock family's story is the most devastating of all the individual accounts. Elizabeth Hancock buried 6 of her children and her husband in just 8 days in August 1666, 7 family members in 8 days, digging every grave herself because no one would come near the bodies out of fear of infection. The field where those graves were dug is still there today, outside IM, and is still called Riley Graves. By November 1666, the outbreak had stopped, likely because the disease had run out of new susceptible hosts in the isolated village, or because cooling temperatures reduced flea activity enough to break the transmission chain. The quarantine had held. Now jump forward 330 years to the 1990s, and a completely different kind of medical mystery. Scientists were studying a question that had puzzled HIV researchers for years. Why did some people, despite repeated exposure to HIV-positive partners and high-risk situations, remain HIV negative? There was a small but consistent group of individuals who seemed to be naturally resistant to the virus, not just lucky, but genuinely protected by something in their biology. In the mid-1990s, researchers identified the answer: a specific genetic mutation, a 32 base pair deletion in the CCR5 gene on chromosome 3, designated CCR5 delta 32. Here is what it does in plain language. The CCR5 protein normally sits on the surface of white blood cells, specifically the CD4 plus T cells and macrophages that HIV needs to infect. HIV uses the CCR5 protein as a doorway, a co-receptor it grabs onto to enter and infect the cell. People with the CCR5 delta 32 mutation produce a broken, shortened version of this protein that never reaches the cell's surface properly. The doorway HIV needs simply does not exist. Individuals who inherit 2 copies of this mutation, one from each parent, are almost completely resistant to infection by HIV-1, the strain responsible for the global AIDS epidemic. People who carry only 1 copy have partial resistance, and, if infected, typically progress to full AIDS significantly more slowly than average. The mutation exists almost exclusively in people of European descent. Its frequency ranges from about 5 to 14% across different European populations, higher in Northern and Eastern Europe, lower in Southern Europe, and essentially absent in East Asian and Sub-Saharan African populations. When researchers used population genetics to estimate when this mutation first appeared and began spreading, the answer that came back from multiple studies was approximately 700 years ago, placing its origin in Europe at almost exactly the time of the Black Death. The Black Death of 1347 to 1353 killed an estimated 30 to 60% of Europe's entire population in its initial outbreak, one of the most catastrophic mortality events in human history. And plague did not visit once. It returned roughly every 10 to 15 years across Europe through the 17th century, with IM's 1665 outbreak being one of the later waves of a disease that had been killing Europeans repeatedly for over 300 years. The hypothesis that emerged from this data is this: if CCR5 delta 32 also provided some protection against Yersinia pestis, the plague bacterium, then every major plague epidemic would have acted as a filter, killing people without the mutation at higher rates and allowing people who carried it to survive, reproduce, and pass it on. Over hundreds of years and dozens of plague outbreaks, that selection pressure could have pushed the mutation's frequency from near zero up to the 5 to 14% range seen across Europe today. Researchers specifically studied the descendants of IM survivors to test this. A study by researchers at the University of Liverpool, published in 2005, found a higher-than-expected frequency of the CCR5 delta 32 mutation in the IM survivor population compared to the general English population, consistent with the idea that carrying the mutation made surviving the 1665 plague more likely. The biological mechanism by which this might work is still being studied. Yersinia pestis does not use CCR5 as a direct entry point the way HIV does. But researchers have proposed that CCR5 plays a role in the immune response to plague, and that disrupting its function may alter the dangerous inflammatory cascade that kills patients with septicemic plague. It is worth noting that a 2003 study by Galvani and Slatkin in the Proceedings of the National Academy of Sciences argued the statistical evidence more strongly supports smallpox, rather than plague, as the primary evolutionary pressure driving CCR5 delta 32 frequency, since smallpox interacts more directly with CCR5-related immune pathways. The true answer may involve multiple diseases applying cumulative selection pressure across several centuries of European epidemic history. But whether the primary driver was plague, smallpox, or a combination of both, the medical consequences of discovering CCR5 delta 32 have been very real and very significant. In 2007, the FDA approved a drug called Maravirac, brand name Cells Entry, the first antiretroviral drug specifically designed to block the CCR5 receptor and prevent HIV from entering cells. It was a direct pharmaceutical exploitation of the same vulnerability in HIV's biology that the CCR5 delta 32 mutation creates naturally. And then there is the case of Timothy Ray Brown. Brown was an HIV-positive American living in Germany who was diagnosed with acute myeloid leukemia in 2006 and needed a bone marrow transplant. His hematologist, Dr. Gero Hutter, had an idea. What if the donor for the transplant was selected specifically from individuals who were homozygous for CCR5 delta 32, naturally resistant to HIV? The transplant went ahead. Brown's existing immune system cells were replaced with cells from a CCR5 delta 32 donor. From the point of that transplant until his death from leukemia in 2020, 14 years later, Timothy Ray Brown's HIV infection was undetectable. He became known publicly as the Berlin patient, the first human being ever documented to have been functionally cured of HIV. A second patient, known as the London patient, and later identified as Adam Castiglio, achieved the same result in 2019 through the same approach. As of the early 2020s, multiple additional cases of apparent HIV cure via CCR5-edited or CCR5 delta 32 bone marrow transplants have been reported, and researchers are now exploring whether CRISPR gene editing tools could replicate this effect at scale, editing CCR5 out of a patient's own cells without needing a matched donor at all. IAM today is a functioning village of approximately 900 residents. The Church of St. Lawrence still has plague death records scratched into the wall plaster. The plague cottages on Church Street still stand, with plaques naming the victims who died in each house. An annual Plague Sunday service is held at Cucklet Delph every August, at the outdoor site where Reverend Mompesson preached to keep his congregation from gathering indoors. The voluntary quarantine of 1666 is cited explicitly in public health literature as one of the earliest and most remarkable examples of community-driven epidemic containment, and was invoked directly during the COVID-19 pandemic of 2020 and 2021 as a model of collective sacrifice for public health. The people of IAM did not know about genes. They did not know about CCR5 or HIV or CRISPR. They made a decision based entirely on a moral calculation. Our deaths, contained here, are better than spreading death to everyone around us. And folded inside the biology of their descendants, in a small deletion in a gene on chromosome 3, is the evidence that they were right, and that their sacrifice left something behind that is still saving lives today. There is a video on this channel that covers the original Black Death of 1347, the initial outbreak that killed up to 60% of Europe, where it came from, how it moved, and what European society looked like in its immediate aftermath. Watch that next, because understanding the scale of that first wave makes everything about IAM and about CCR5 delta 32 make far more sense.